Viruses are mysterious entities. Microscopic, much smaller than bacteria, and not truly alive by most definitions. They can't reproduce on their own. They need a living cell to multiply. Coronaviruses are a large family of these viruses, named for their crown-like spikes seen under electron microscopes. These spikes aren't just for show. They're the keys the virus uses to unlock and enter our cells. SARS-CoV-2, the virus behind COVID-19, is one such coronavirus. Coronaviruses aren't new. Some cause mild colds, but others, like SARS, MERS, and now COVID-19, can be far more dangerous. They have the potential to cause global health crises. Understanding their structure is crucial. They're essentially packages of RNA genetic material, wrapped in a protein shell and those signature spikes. Their only goal is to replicate, but they can't do it alone. They must invade a host cell. Without a host, they're inert, waiting for a chance to spring into action. Once inside, they hijack the cell's machinery to make more copies of themselves. This is the fundamental nature of a coronavirus, a microscopic invader using our own biology against us. Their genetic material is RNA, not DNA, and this difference shapes how they operate. The spikes are what make them so effective at infecting humans. These features are why coronaviruses can adapt and sometimes jump from animals to people. Their ability to spread and evolve makes them a persistent threat. That's why understanding them is the first step in fighting the diseases they cause. Coronaviruses enter our bodies mainly through the nose and mouth, sometimes the eyes. They travel in droplets or aerosols released when an infected person breathes, talks, coughs, or sneezes. Once inside, the virus targets specific cells using its spike proteins. For SARS-CoV-2, the spike protein locks onto the ACE2 receptor, found on cells in our lungs and airways. This is why COVID-19 primarily affects the respiratory system. The spike protein fits the ACE2 receptor like a key in a lock, allowing the virus to enter the cell. The cell membrane engulfs the virus and the viral RNA is released inside. The cell, tricked by this interaction, unknowingly welcomes the invader. The viral RNA then takes over, giving instructions to hijack the cell's machinery. The cell becomes a factory for making new viruses. This Trojan horse strategy is what makes coronaviruses so effective. The stage is now set for the virus to replicate and spread. Infection has officially begun. Once inside, the coronavirus's RNA blueprint takes over the host cell. The cell's ribosomes, which normally make proteins for the body, are hijacked to produce viral proteins instead. The viral RNA is copied thousands of times, and new structural proteins are made. These include the spike, envelope, and nucleocapsid proteins. The cell's own machinery is now dedicated to assembling new virus particles. The endoplasmic reticulum and Golgi apparatus become virus factories. New virus particles, or virions, are assembled and packaged. The host cell fills with these new viruses until it's exhausted or destroyed. The viruses escape, either by budding out of the cell or by bursting it open. Thousands of new viruses are released, ready to infect more cells or be expelled to infect others. This rapid replication is why infections can spread so quickly. The host cell is left damaged or dead, while the virus continues its cycle. Each new virus is a clone, ready to repeat the process. This is how a single infection can quickly escalate. Think of the virus as a rogue software programmer with a USB stick. It's RNA. Your cell is a computer, running essential programs. The virus finds a USB port, the ACE2 receptor, and plugs in its code. The cell's processor, the ribosome, starts running the virus's instructions instead of its own. The first command, make thousands of copies of the viral code. Next, build casings for these codes, viral proteins. The cell's resources are hijacked, producing new viruses instead of normal proteins. Soon, the cell is overwhelmed, its original functions lost. Eventually, the cell breaks down, releasing all the new viruses. These viruses then seek out other cells to infect. The process is like a factory taken over by an industrial spy, churning out only viral products. This is how the virus multiplies rapidly inside us. Our own cells become unwitting accomplices in the spread. Once new viruses are released from infected cells, they spread through the air. Coughing, sneezing, talking, or even breathing can expel virus-laden droplets and aerosols. Larger droplets travel short distances and may land on surfaces or people nearby. Aerosols are lighter, lingering in the air for minutes or hours, especially indoors. This is why close contact and poor ventilation increase transmission risk. The virus can also survive on surfaces for a time, though this is a less common route. Touching a contaminated surface and then your face can transfer the virus. Hand washing breaks this chain of transmission. Airborne spread is the main driver, especially in crowded indoor spaces. People are most infectious just before and during the early days of symptoms. Some individuals, called superspreaders, can infect many others, often in crowded events. Viral load, the amount of virus someone sheds, matters for how contagious they are. Understanding these routes helps us block the virus's path. Strategies like ventilation, distancing, and masks are based on this knowledge. Stopping the spread starts with understanding how it moves. Scientists moved fast to identify SARS-CoV-2 in late 2019. Using genetic sequencing, they quickly decoded its RNA blueprint and confirmed it was a new coronavirus. This information was shared worldwide, accelerating research and response. Electron microscopes revealed the virus's crown of spikes, confirming its identity. Lab experiments showed how it infects and damages cells. Epidemiologists traced outbreaks, identifying high-risk activities and vulnerable groups. Data on symptoms and outcomes shaped public health guidance. Mathematical models predicted the pandemic's course and the impact of interventions. Rapid diagnostic tests like PCR tests and antigen tests were developed to detect infections. Wastewater surveillance emerged as an early warning tool for rising cases. These scientific advances have been crucial in our fight against COVID-19. By 2025, our understanding of COVID-19 keeps evolving, just like the virus itself. SARS-CoV-2 mutates, creating new variants that can spread faster or evade immunity. Scientists track these changes with constant genomic sequencing, updating vaccines and treatments as needed. Long COVID remains a major research focus, with persistent symptoms affecting many. Researchers are uncovering its causes, from chronic inflammation to immune responses. Diagnostic tools are improving, with faster tests and better surveillance, including wastewater monitoring. Predictive models help public health agencies prepare for future waves or new pathogens. COVID-19 is now endemic, like the flu, requiring ongoing management rather than emergency response. Maintaining population immunity through vaccination and boosters is key. Protecting vulnerable groups and having rapid response systems are essential. Our scientific journey with this virus continues, bringing new insights each year. Living with COVID-19 means adapting, not becoming complacent. Ongoing vigilance and innovation are our best tools. Even in 2025, basic precautions remain vital. Vaccination is our strongest defense. Updated boosters help our immune system recognize and fight the virus. Staying current with recommended doses is especially important for vulnerable groups. Good hand hygiene, washing hands thoroughly or using sanitizer, removes virus particles. Avoid touching your face to prevent transferring the virus from surfaces. Improving ventilation in indoor spaces dilutes airborne virus, making environments safer. Open windows, use air purifiers, and ensure good airflow in shared spaces. Mask wearing still matters, especially in crowded or poorly ventilated areas. High-quality, well-fitting masks add a layer of protection. If you're feeling unwell, wear a mask to protect others. Consider your own risk and those around you. These steps work together to reduce transmission. Simple actions make a big difference. Stopping COVID-19 is a shared responsibility. If you feel sick, stay home to avoid spreading the virus. Get tested if you have symptoms. Knowing your status helps protect others. Follow public health advice, including isolation if needed. Support public health initiatives and stay open to new recommendations as science evolves. Trust credible sources. Misinformation can undermine efforts and create confusion. An informed public makes safer choices and builds community resilience. Global cooperation is crucial. Viruses don't respect borders. Supporting vaccine access and sharing knowledge worldwide protects everyone. Uncontrolled spread anywhere can lead to new variants everywhere. Investing in public health infrastructure and surveillance prepares us for future threats. Promoting health equity ensures no one is left behind. By working together, we can manage COVID-19 now and be ready for what comes next. Our collective actions shape the future of public health.